Hello, I'd like to start today by thanking the Beckman Foundation, not only for the opportunity to give this seminar in their annual meeting, but also just for the support that they've given to me personally and to the field over the years with centers, the cryo-EM centers and the light sheet centers that I will be talking a little bit about what we're doing at Washington University today. It's very important that philanthropic organizations, the Beckman Foundation, step up to create these new opportunities for new technologies to be applied to biology, something that's very difficult to fund through other mechanisms. So I'm very happy to be here and very happy to have one of these light sheet grants from the Beckman Foundation. So the motivation for the work that I'm going to tell you a little bit about today, to just have a scaffold for what we're doing, is working on diabetes-related glucose-stimulated insulin secretion, glucose homeostasis in the body. And this is my blood glucose for my height, weight, and age, about 4.6 grams. This is how much glucose I should have circulating in my blood at any given time. But it's really easy to add almost a 10-fold bolus. And probably many of us have done this on a hot day when you're really thirsty. You just slam a can of Coke. Well, you've actually all of a sudden doubled, 10-fold increase in the amount of glucose that you should have in your resting blood. And so what is amazing to me is not that we have a diabetic of diabetes in this country or in the world, but that we aren't already all dead of diabetes. And the reason is it's amazing little microorgans, the pancreatic islet or the islet of Langerhans. These are small microorgans, about 100 microns in diameter, 1,000 to 10,000 cells. And they contain the insulin secreting beta cells, but also the glucagon secreting alpha cells and the delta cells that secrete somatostatin that work as a regulator with the other two cell types to keep islet function correct. And here's an image of a human pancreatic islet. And you can see that most of the cells are the beta cells. In humans, about 65%. There's a few alpha cells, around 10 to 20%, depending on the species, and a few delta cells. And this is a section of several 3D sections that have been merged together in a through focused view just to show you the cells. But as I said, these are about 100 microns in diameter. And what happens is when there's an increase in blood glucose, the islets secrete insulin, which tells the muscle and adipose tissue and the liver to start taking glucose up out of the system and storing it. And then, of course, that restores normal glycemia. And when there's a hypoglycemic episode, then there's a decrease in blood glucose. At the same time, then insulin is shut off and glucagon secretion increases, mainly telling the liver to liberate stored glucose, but also telling the rest of the body to stop taking up glucose, to keep it in the circulation. The brain needs glucose to survive. Hypoglycemic unawareness and sudden death of patients with diabetes is the biggest fear, that somehow glucagon secretion and insulin secretion will not be correct to actually liberate enough stored glucose to restore euglycemia. So this is what we're interested in studying, and we're interested in studying these cell types in their native environment, because if you take them out separately, they don't have the same behavior. Here's a beta cell, and this canonical model of glucose-stimulated insulin secretion, where glucose comes in, shunted through glycolysis and citric acid cycle, leading to a change in ATP-ADP ratio, closes these potassium channels, depolarizing the membrane, opening voltage-gated calcium channels. Calcium comes in, that triggers insulin secretion, very much like any synaptic secretion that maybe people are more familiar with. Also opens these other potassium channels that are calcium sensitive, which repolarizes the membrane. And they get these cycles of membrane depolarization, repolarization, depolarization, repolarization. With about, well, they vary over time, but at first they can be maybe 10 to 20 seconds. And these also lead to increases in calcium, as every time the membrane depolarizes, there's an influx of calcium, so you get these calcium pulses. And these correlate very well with pulses of insulin secretion that come out every time the calcium goes high. Well, we'd love to measure insulin secretion directly, and we work on ways to do that. But one thing we do know how to measure very nicely in a microscope is calcium. But we now need to measure calcium in these cells in this 100 micron thick, gathering of cells, sort of an organoid of cells, the pancreatic islet. And how we've taken to doing that to be fast enough is to use light sheet microscopy. And in particular, we've been using the dual view light sheet that was invented by Harry Schroff's lab at NIH, I guess now almost coming up on 10 years ago. And we've used this, and this is the system we built in our lab to image islets. And what it does is you have one light sheet that comes in and you look from 90 degrees, and then you turn it around and you put the light sheet through the B channel and you look through the A channel. By combining these two views, you no longer have a Z view. You have 2X, 2Y, and 2Zs, but you throw the Zs away, allowing you to use a thick light sheet and still get high resolution throughout your sample. And an example is shown here. These are alpha cells that have been labeled with the calcium indicator GCAMP6F. And you can see that in just a single view, you can see the cells. The XY resolution actually looks pretty good, but you have a lot of haze because you've used this thick light sheet to go through, and you aren't really rejecting that with a confocal pinhole or anything. And the light sheet is Gaussian, so there's a lot of spread off of not just at the middle, but out into the wings. When you combine the two views, you see the XY resolution doesn't change that much. You can see if you look at, say, this cell here, it sort of looks similar. These cells, different places, look somewhat similar, but that the background is really suppressed because now you have high resolution, not only in X and Y, but also in Z. And using the 40x.8 NA lenses that we're able to use, we get a resolution about 300 nanometers in every direction. This allows us to go very fast. This is taken with 50 milliseconds per frame, 60 images per stack, 1 micron step size, and that gives us a total volume time of 3 seconds. So every 3 seconds, we're taking a full 3D volume of the islet through 60 microns. We can go a little faster with the signal to noise. We can go a little larger frames, larger images, smaller image stacks, depending on what we want to do. But this is quite convenient to see many, many of the cells in the islet and how they talk to each other. Now that's what we're really interested in. I showed you before that glucose-stimulated insulin secretion is a canonical pathway that's pretty well understood, but it's really half the story. There's all kinds of other ways. There's the brain influences insulin secretion. The gut influences insulin secretion. There's several hundred GPCRs that are expressed in the islet, not only in beta cells, but also in the alpha cells and in the delta cells. And these cells all talk to each other. And what we want to know is how do all of these communication pathways, all of these millions of possible communication pathways, which ones are important? Which ones can we modulate to ameliorate diabetes? Which ones can we actually stimulate, perhaps to completely cure diabetes, for instance? And so we want to look at these pathways, and we want to look at them all at the same time. At the same time as we've learned more and more about the complication of the pathways, is broad spectrum of fluorescent proteins have been developed. And so we're not stuck just with using GCAMP6 anymore, which would be sort of this greenish, it's sort of GFP derived. But there's also yellow and cyan and blue and orange and red and deeper red and deeper red. And there's also IR variants I haven't shown here that have been developed. And so there's a broad range of spectral components that we can use, but you see these are all highly overlapping. One of the things about the fluorescent proteins is they have these long tails, and they're somewhat wide sometimes in their spectral width. And so they are not necessarily the best behaved spectrally. And so it's really hard to say, take a filter and filter out the blue from the cyan, from the green. You can see that filtering out the orange from the red, well, the orange is pretty big all through there, right? It's a little bit brighter. And so how do those things go together? How do those things that are overlapping, how can we tell them together in a measurement and separate them? And so what we've been trying to do is to use spectral imaging methods or hyperspectral imaging methods. Typically what people have done is point scanning. So Zeiss has their microscopes, originally the Meta and now 880, 980 quasar detectors, where you take a point scan, and at every point in that raster scan, you take a spectra. There's also ones where you do serial filters and you step through the filters, or you can do a split scanning, which is another line scan type of approach where you took an X lambda picture and then you scan that over Y. And all of these are possible, but they're slow. And we want to be able to watch these 10-second oscillations over the entire islet at maybe 1 second per, or a couple of seconds, so that we can resolve what's happening at the different times in different cells. What we really want to do is collect all this cube at once. So we want to take the X, Y, lambda cube all at once and then scan that over Z, or even take an X, Y, Z lambda cube eventually all at the same time and then repeat those over time. So what we've taken is an approach called image mapping spectroscopy that allows us to take an X and a Y and lambda, the entire cube, all at a single time, and then step that through Z. This is really nice with the light sheet, because if we can take a single light sheet, we can move the light sheet up and down in Z, and we can then collect a Z stack, and then repeat that over time to have an eventual X, Y, Z, lambda, T, 5-dimensional stack of data. So how image mapping works is, here's our image of different colors. This is the object, and this is an intermediate image that have all these different colors. And we use an image mapper, which is a multifaceted mirror, where the first line of the image at diffraction resolution is put to one place into another intermediate image. The second line is placed to another place, third line to another place, fourth line, etc. And there's blank regions left in between. This intermediate image is then run through a prism, which spreads all of the spatial spaces out in lambda, in wavelength, such that this end result is a 2-dimensional picture. Every pixel corresponds to one X, one Y, and one lambda. This gives us then a simple remapping to make a data cube that is X, Y, lambda data cube. We can then repeat this over Z to make the X, Y, Z lambda, and repeat that over time to get our 5-dimensional image that we want. Now you can see that if I want to take a 512 by 512 image and take 30 colors, well, I no longer can just do that on a small camera. But fortunately, large frame cameras have come out. They've become very readily available, mainly because the cell phone industry uses large format CMOS cameras. Scientific grade cameras have come out, certainly exceeding 10 megapixels. Although the data I'll show you today was done with 5 megapixel camera, which limited us to something like 300 by 300 X and Y by about 30 lambdas. Now, here's our human islet again, as I told you. And these cells are not only, just neighbors, they are all intimately connected with many chemicals that go back and forth. And I don't have time to go through all of these, but I'm just going to tell you 2 of the ones that we know the beta cell secretes that affect the alpha cell. One of those is insulin, which of course is well known. The other is zinc. And zinc also is secreted from the beta cells, co-secreted with insulin, and it does affect glucagon secretion quite dramatically. And people aren't sure why. The alpha cells also form a very heterogeneous population, and there's been some data, I'll show you some data here, and there's also been more recent data really showing that zinc handling in the alpha cell seems to correlate with the different subpopulations of alpha cell that generate the heterogeneous population that we see. Well, here's what we did to do this experiment, just as an example, is this was just all done at 488 nanometer excitation, so our standard blue light, again with our 40x.8 NA diSPIM objectives. We measured zinc using Fluorescent, which is green, calcium using calcium orange, which is orange, and that's shown here in the yellow, which is maybe a little bit difficult to see on this slide. And then the alpha cells or beta cells were labeled with a tandem dimer RFP using transgenic mice, and that's shown here. And so you can see that this TDRFP and the calcium orange peak, they overlap completely. But if we're able to take the entire spectrum, they're quite different. It's easy to tell something that has this reddish broad spectrum versus something that has the narrower orange spectrum, especially doing when you do a hyperspectral image and a linear deconvolution, linear unmixing or single value deconvolution. And here's the 3 different colors that have been separated out of a single islet. In this case, the TDRFP is in the alpha cells. The calcium orange is in all the cells because it's just loaded in, and the Fluorescent, the same thing. It goes to all the cells because it's loaded in as an external dye. So we load these in as AM. Acetoxymethyl ester forms the dye. They're cleaved inside, very much the way calcium and zinc imaging went for many, many years before fluorescent proteins came on the scene. So here's the beta cell quantitation of calcium and zinc. And as we switch from low glucose to high glucose, we see these calcium oscillations that are really the hallmark of beta cell function. And we see that there's a decrease in zinc. This is perfectly normal. The zinc is co-secreted with the insulin, and it's not brought in as fast as to replenish. And so over time, we see the zinc goes down. And the longer we allow these calcium transients to go, they'll go down, and it'll go down to about 40 or 50%, and then it'll sort of stay.
down there for a while. Sometimes it goes down a little lower, and that feeds back into actually disrupting the calcium oscillations in the long term. But this is exactly what we see, what we expect to see. And that was known before we did these experiments. We then turned our attention to the alpha cells, where we have the labeled alpha cells. And here we start to see these different heterogeneous populations. There's alpha cell 1, when we go from low glucose to high glucose, we see the calcium goes up. Even though we know the glucagon secretion goes down, in that one, the calcium goes up. But what happens to the zinc? The zinc in that one, nothing happens. It's not taking up any zinc, but its calcium is going up. If we look at another cell, cell 2, the zinc isn't, the zinc doesn't changing very much at all. But what's happening to the zinc? The zinc goes up. So somehow, if the zinc is being incorporated into the alpha cell, that is decreasing the calcium, somehow keeping it clamped off or decreasing calcium. And so it may be that there's a link between the zinc coming in and the Ca2 channels that are required for depolarizing the cells and the beta cells, and also are active in a not quite fully understood way in the alpha cells. But the hypothesis coming from just this single experiment, all we did was look, is the Ca2 opens, that closes the calcium channels, and somehow that keeps calcium in the second cell where zinc is high, one with the stars, keeps the calcium turned off. So what I've shown you today is really a way we do multicolor, single snapshots. We have great sensitivity of the IMS to distinguish these multiple indicators. We get simultaneous acquisition of both spatial and spectral properties, and we can go very fast. With a 10 megapixel camera, we can do 512 by 512 X and Y by 36 lambda. This is probably good enough to do many of the experiments we want to do. The drawbacks are the data sets are huge. This is 3.4 gigabytes per second comes out of these cameras, out of the two cameras for the diSPIM. Easily generate 2 terabytes per data set. Even if we skimped a little bit, 1 terabyte data set, that's pretty big. And I think the technical term we use is this is hard to deal with. So fortunately, the Beckman Foundation has given some money to us and other centers to try to address these problems. This is the hardware that we have. We've purchased a lattice light sheet with the Beckman money, along with matching funds from Washington University. We had an existing Zeiss LSM1 and the diSPIM I showed you. We've been able to buy some new high performance computing to allow us to deal with the computations that we need to do. And we've bought new research storage, about one petabyte of additional storage in both the high performance active and archive tiers to supplement what we already had in our cell imaging center, which also has to share with cryo-EM and other data intensive measurements. So what we really want to do is build the pipeline for a seamless way to analyze all the data. And this is built around data fusion, feature extraction, and data analysis. It's all AI routines that are hidden from the end users. The pipeline is written in Julia, which is an open access programming language, which allows us to do high performance, multi-terabyte data files can be used very easily with on-demand computation, and it's arbitrarily written for single code writing, can be scaled to an arbitrary number of dimensions, which becomes very useful when you get to X, Y, Z, lambda, and T. The data fusion, well, we have reconstruction, registration, and stitching. Again, we're exploiting deep learning to take the natural redundancy that comes in spatial and temporal and spectral correlations to help stitch the data together to register data, say from the two views. And this is, we'll all have pre-trained deep learning models for advanced imaging tasks. The feature extraction, of course, we need to scale this to massive data sets. Oftentimes, if all you're interested in is the number of cells or what a cell is doing, you can reduce the complexity of the data to something really small, and then you can apply all of the data analysis you've already had. Again, smart allocation of storage to the most relevant data is what we're going with on that. And again, pre-trained models for users with a user-friendly GUI, which we think is going to be highly important to get this in the hands of as many biological researchers as possible. So that's where we're going, and the reason, the motivation. I just want to thank the people in my lab, Ca2 and Zn2 now has his own lab in Milan. Italy, Amy Elliott, who's a postdoc at NIH, and Alessandro, who's a senior scientist in my lab still, who have been the ones that contributed to this. Our collaborators from Rice University who developed the image mapping spectroscopy, funding from the NIH. And then of course, none of this would be possible, what we're planning to do and what we're doing for helping the entire field with the software through the Beckman Light Sheet Microscopy Center, my co-PIs, James and Tim, and Beck Kameloff, who's our computer scientist, and of course the funding from the Beckman Foundation. And so thank you very much for your attention.